Okay, so let me sort of continue. So we got to the point where we sort of saw the good and the bad in the sort of the classical. Let me put it this way. You know, the original David Reddish suggestion for why cocaine becomes addictive, right? Or why you learn to choose and continue to compulsively take cocaine in spite of anything else that happens, all right? And so some of these, okay, so some of these you might say, well, you know, you don't know, like the blocking thing, the quality of the paper that showed it may be questionable. But other things are definitely fairly catastrophic to this model, and that is particularly this value goes to infinity and et cetera, right? So, you know, and basically everything is around this equation where you have this max operation also is kind of implementationally for a neural circuit. It is not completely evident how you would perform this sort of an operation, right? It's not a subtraction like excitation minus inhibition, right? It's not a division. It really is a kind of a, it's not even a threshold. No, it is a thresholding operation. Okay. All right. Okay, and so the crux of the proposal is that, you know, dopamine activity never converges to zero, right? Dopamine activity codes error signal. Error signal never converges to zero, and so you get this overvaluation of drug consumption state, okay? All right? But the thing is that this value goes to infinity, so is there something that we can sort of, we can augment or change our reinforcement learning algorithm to deal with some of these problems? Note the important thing is that if you change this reinforcement learning algorithm, you better show that the new algorithm that you have is also leading to optimal behavior, right? What I didn't have time to show you is that if you take your reinforcement learning algorithm, you can show that it will lead to economically rational behavior, meaning that you will learn to seek rewards, you will learn action policies that maximize possible rewards. So it's a normative model, okay? So now if we're going to screw up with our reinforcement learning algorithm, we better not screw up the normativity, right? Otherwise it's just ad hoc, it has less value conceptually. Okay, so you know, so how could we, you know, so one way to sort of to fix some parts of the original proposal is to say now that instead of, what if instead of discounting our error in time, for example, or discounting our rewards in time and calculating the error as depending only on the reward received and the reward expected in a particular state and action pair, what if the error also depends on the average or mean expected reward in a particular environment? Okay? All right, so this is called average reward reinforcement learning. You can show that if you define the error like that and include it in your Q update equation, that will still lead to optimal behavior, right? So it's still a normative model, but notice it's a different model for learning, right? It says that, you know, all of your learning is pithed against how much is your environment rewarding in its total, right? Okay, so in the sense that the more the Q's go up for all state action pairs, right, the more this average reward R increases and the more strict this delta equation becomes, right? Yeah, so you need much more reward to drive it as the R bar goes up, okay? So then what you can say is that now this is going to be our natural reward error, okay? You can now say that this is going to be our cocaine reward error. Notice it's exactly the same as in the Reddish model, except for this average reward term. Okay, all right, so if you include that, and actually this was work that was published by a really great group of, at that time, undergraduates in Tehran, Disfuli, Karamati, and Pire, I think. So what under under these conditions, under this learning signal, what you can show is that the error signal goes down for the natural rewards, but also for the cocaine choice. This is the solid is the cocaine choice, right? And it goes down to 0. Okay, so also the cocaine choice error is learned away. Okay, right? The value for the cocaine choice asymptotes. All right. Okay. All right. Okay, so notice that this is total reward error, despite the fact that we still keep this, pharmacological effect, right? We haven't taken that away. But the reason why it goes away is that if you keep going up with the Qs all the time, right, this R increases and eventually it will learn it away appropriately. Okay? All right? And now we can also include another term, right? And that term, now it's sort of ad hoc, sort of heuristic, is to say that when you're exposed to dopamine, right, there is another slow process, right, that every time you take dopamine, it will directly augment this average reward by some inappropriate, additional term, kappa, right? And so now we can redefine this error using the rho instead of R, right? Okay, all right? And we can simulate the decision processes that are driven now by this learning signal, okay? All right, this is what Disfuli and colleagues did. So first of all, what they showed is that, okay, under just a simple drug delivery schedule, this algorithm will lead to the fact that drugs will win, okay? But they will not win unconditionally always, okay? So first of all, they showed that if you pair the drug delivery, right, with a punishment, okay, all right, what happens is that in order for the drug choice to win significantly over the non-drug choice, you need to have an extended exposure to the drug, right? So once this rho changes sufficiently, right, the reinforcing ability of the drug will increase, and so you will get this increase in responding for the drug lever, for the lever press with the drug reward, despite the fact that you're being punished to it. So you sort of get resistance to punishment with long-term exposure in this model. Sorry.
Which parameter determines this interval until they actually increase the probability of selecting?
Probably this K, right? Well, what I should really put all the equations. There is also a kind of a time constant by which. So if this K is by how much you augment this average reward variable on each drug delivery, right? So if it's small, it takes you many visits. Okay, so. I mean, they were not, at that time, they were not concerned, well, they were not necessarily concerned with quantitative modeling. What they wanted to show is that taking this into account can give you these effects, that it's the long-term exposure to the drug that makes you compulsive, meaning that you take the drug independent of the fact that you're receiving punishments. But under short exposure, you would be sensitive to it. Okay?
I'm asking because when we did our experiments with introducing the punishment, we also had many animals that would go down for some time and then they come back up. So
yeah,
is that
see, it's go down for some time, then goes back up. Okay. Yeah, okay. So this is the short term, learning it away because you're being punished, right? But then it doesn't go to 0, so you still take a bit, and then this row builds up, and then you go back up, okay? All right? So you can also model impulsivity or this sort of proximal reward choice. So in this state space, what happens is that before you get the reward, you need to go through many other states, and that is a time-dependent process, right? And only at the end of this delay, you get the reward, natural reward, large, sort of delayed reward, and this would, and this would be relatively small reward, let's say, even if it's small, so the drug reward, right, you would go for the proximal small reward as opposed to the delayed long reward, and that develops after a certain period of time of being exposed. So this is the probability of selecting C2. Hold on, how does it go? It should go down. Okay, this is the drug choice. It goes down below the non-drug choice. Okay, all right. So the other thing that this, let me see what's next. Okay, the other thing that this model did reflect, and that is that you can get blocking for the drug effects under long-term exposure to the drug, right, and no blocking under short-term exposure to the drug, which appears to be consistent with some of the data, all right? And also, because the error goes to 0, as I showed you before, right? The errors goes to 0, the values for the drug also asymptote, so there's none of this question of how do you encode value of infinity, okay? So it appeared like it's a promising manipulation of this model, right? And what it says basically is that there is some long-term slow process, right, that modulates this reward signal error. And this long-term slow process is sufficient to fix some of the ails of the original proposal. But it's still positive reinforcement for the drug, right? So you learn the drug behavior because of its positive reinforcing qualities. Okay? So another interesting proposal. So, okay. This was a slide basically to show you an example of the fact that if you measure dopamine outflow, and Garrett, please don't kill me, but help me maybe a little bit. You might know this much better than I do. Okay? And you measure the outflow of dopamine in the ventromedial stratum or dorsolateral stratum. What you see is that when you expose the animal, so it's a self-administration of cocaine task, I believe. If you let the animal develop, go through administration exposure for a long time, at the short time you see sort of a large, strong dopamine signaling in the ventromedial stratum, virtually none in dorsolateral stratum. Okay, and then what happens is that the signal diminishes. There is a bit at medium time. There is a sort of a development of signaling in the dorsolateral stratum. Okay, and this is I believe a rat going through an escalation paradigm, right? And so there's something very complicated going on, right? When you put an animal through extended access to the drug, right, where it starts to escalate its behavior, okay? So there's something more than just, static reinforcement error that never goes away because of the drug action, right? And also it's interesting that now a different piece of data that I also pulled out, this is from Takahashi's paper. This figure is from Frontiers, but the Frontiers paper was about another publication where they recorded in the ventral striatum, and they also recorded in the dorsal stratum, and this was a mouse, I think, going through a go, no-go paradigm where the nose poke led to cocaine persistently, all right? And they recorded from neurons that a priori were selective for a particular stimulus versus another, right? So they were selective, they were enabled to encode the sort of the stimuli and the outcomes of particular actions. And after exposure to cocaine, this selectivity went away, right? Okay, and so something is happening not only about the input, let's say to the striatum, to the ventromedial striatum, but something is happening about the ability of the neurons in the ventral striatum to encode information, right? Which could be values associated with particular stimuli, right? Or it could be stimulus outcome contingencies, or Lord knows what, okay? And so the question was that, well, how could you put at least some of this into a reinforcement learning framework to start understanding what the behavioral consequences could be of these changes, all right? And that's was a quite neat proposal from Jeff Schoenbaum and Yael Niv. And what they proposed, in fact, is that what's going on here is a drug-induced impairment of the critic in the actor-critic version of the reinforcement learning algorithm. And it's the following, right? So under normal conditions, you have this dopamine error signal that that drives the learning in the critic, and also, instructs, and these values, right, instruct the actor, right? And these weights of associations, as I was telling you, associations between stimuli and actions, right, are also driven by the same error signal, right? So for the actor in a normal condition to do the right thing, right, the values of the states have to be learned correctly, right? And then the weights between the values and the actions have to be learned, okay? All right, so you have these two equations, essentially, okay? All right, and so what happens, they suggested what happens with the drug, and that is the learning in the critic is somehow attained. I mean, it's impaired, okay? So the critic doesn't learn, but the actor keeps on learning, right? So that would be compatible with, if we think that the critic is somewhere in the ventral parts of the striatum and the actor is in the dorsal parts of the striatum, right? This idea would be compatible with dopamine signaling decreased in the ventral striatal structures, right? But not particularly touched in the dorsal striatal structures. Yeah? So then in this case, okay, this error here, right, drives the action update, okay? But notice the difference, okay? Here, the value update and the action update depends on the values for the S's that you have learned, and you always update them. Okay, sorry. Yeah, okay. So the update of the action plan depends on the values that you also learned, right? So that's the interplay, okay? When the critic can't learn anymore, then this delta, right, depends on the initial value of some S, right, minus the initial value of some S, because it doesn't change, plus the reward, right, which equals to just the reward, right? So then the actions that you learn, the action plans that you learn, depend only on the immediate reward, okay? And they don't depend on the estimate of how much you might get if you take that action plan in the future, right? So what happens in this situation, right? First of all, since these guys are not learned, right, then if you start out in the situation where all the stimuli and all the actions are predicted to be equally valuable, right, if these values are signaled in the ventral striatum, you're going to see that the selectivity of your neurons will be zilch, nothing. They will fire equally for all the stimuli, right? And the second is that if you take this agent now and you simulate it, what you will see is it's extremely myopic to anything that happens in the future, right? It's like the ultimate discounter, right? Its behavior is driven only by what it gets now, right? And if there is a punishment associated with it later, it won't take into account. Yeah.
And this transition from ventral to dorsal striatum, is that, I mean, how did you get there? I mean, is this something you...
Okay, so in this proposal, it is not explicitly modeled as a process.
Okay.
It just says, if we were to take this loop out, but
then
leave this loop this, you get that. And if you now go through the logic of this kind of a model, you get a lot of the behavioral phenomenology that you would get. Okay?
Your assignment of ventral and dorsal for the moment is arbitrary.
Well, ventral, it says, you know, these are, there is some evidence to suggest that, you know, the valuation of states appears to be correlated with activity in the ventral parts of the striatum. This comes from imaging studies in humans, for example. Jeff Schoenbaum also has some evidence to that. You know, and dorsal striatal structures and associated dorsocortical loops tend to be more involved in motor plan preparation, motor plans, et cetera, but not state valuation. Okay? So this, another way to think about it, this is the guy that's responsible for Pavlovian learning, right? Because Pavlovian learning doesn't require actions or, you know, explicitly structured actions, right? This is the guy that's necessary for any kind of instrumental learning, right? Why? Because now you're making state action contingencies. You're learning new actions, or you're learning to tie stimuli and actions. Okay? All right. So, and this was, you know, I thought this was an interesting suggestion. I don't know if they're following up on this sort of suggestion with more explicit modeling. I think, you know, this Frontiers paper was an interpretation paper. They said, look, we have this data, and here's how we could imagine it as a model. Okay? All right. So what's next? Oh, yeah. And so this transfer from ventral to dorsal structures, you know, harkens to a theory for transition to addiction advanced by Barry Everett and Trevor Robbins. I'm sure you will see this, maybe some version of this slide when Trevor will come in a couple of days, and luckily I won't be there, so he can't kill me for bastardizing his work. But basically, you know, they focused on sort of functionally and anatomically suggested loops between ventral striatocortical structures and the dorsal striatocortical structures, and they were suggesting that transition between motivated outcome-based behavior, which is normal, right, which is ventral, to kind of habit-based behavior, which is not explicitly tied to outcomes. So dorsal is this drug-induced, you know, transfer of control from ventral to dorsal structures, which is exactly what this actor-critic, this impaired critic idea suggests. Yes, question?
The transition from ventral to dorsal, as far as I understand, it's normally seen when you transition from goal-oriented to habit. So in the end,
yeah,
is it normal?
Yeah. Yeah, that's why I say it's sort of evocative of, and it's not a model of, right? Okay, it's in the same sort of sense, right? We actually did a model that tried to model these loops and the transitions, hierarchical reinforcement learning model, where you had, you know, general goal loops down to specific action sort of valuation loops, and you could show how with the drug you get this transfer of accumulation of error at the bottom of the hierarchy, and you learn away the error at the top. Well, but I don't have time to talk about this. So yes.
So in the actor-critic model, where the critic is the sort of pathological component, and then you get this model for sort of addiction, has anyone sort of hypothesized the opposite way around, where you might have the actor as a pathological component, and would that potentially model other conditions where there are issues with sort of contextual learning and action implementations?
You mean the actor doesn't learn?
Yeah, the actor doesn't learn.
Let's imagine what happens if the actor doesn't learn.
Yeah.
Okay? All right? So if the actor doesn't learn, Okay, so let's say the critic will learn, right? So you will get normal updates of stimuli values and state values, okay? That's all and good. So if you put your animal through Pavlovian task, you will see that it appropriately modifies its Pavlovian response to drug cues, for example. You know, I don't know what would be like a typical Pavlovian response to a drug cue. Approach. Okay, so it runs faster towards the cue. Okay, all right. And if you contingency for the animal is to run away from the cue to get the drug, it will never learn it anyway. Okay, but under that case, your impaired actor animal will never be able to have any kind of instrumental conditioning. Right? It'll do instrumentally, it will do exactly the same thing as it did before. Okay, so now if you imagine that this develops over time, okay, all right, so that potentially could, explain things like instrumental compulsivity, right? But Pavlovian normal responding, okay? All right, I don't quite know how applicable that framework would be. Yes, Marina?
I guess my questions about the loop, if you go to the next slide.
Yeah.
Is those represent their anatomical pathways that connect these regions.
Yeah.
And they don't not exist before drug exposure and exist after.
Yeah.
So the temporal dimension of this has always been fuzzy to me. So the shell is involved in some very early responses when it's just, the first time I have a drug and it's reinforcing.
Mm-hmm.
And the dorsal striatum can be shown to play a very important role after overtreatment.
Yeah,
right?
yeah.
But there doesn't seem to be much to support spiraling transitions, even though there is the spiraling anatomical connection.
Yeah, that's a very good question. You can ask it from Trevor.
I guess the idea would have been that there are stage forms of plasticity that start in the more ventral parts and then
potentially.
More dorsal parts.
Potentially.
Of course, I don't think that experimentally has this really been fully demonstrated. Even if you have the connectivity in the beginning, you can still imagine that the plasticity occurs as a wave.
Okay, so gosh, I wish I included these slides, but we imagine that what happens is that you have a hierarchy of decision processes, reinforcement decision processes, where at the top of the hierarchy you have a general decision process that basically says stay here or leave. Okay, that's an overall goal process, and it only has two states: stay or leave. Okay, under that sits the next level, which would be to leave. I need to proceed here, turn, proceed there, turn, exit through the door, and so that has several steps on it. Okay, and then you can keep on breaking down this hierarchy to basic action plans, right? Basic action decisions, right? Because at each step of the descending down the hierarchy, there are more and more and more steps. So more and more states or state action pairs to learn, right? Even though the signal that you get, the learning signal, impinges upon all of these levels of hierarchy, right? It will take less time to correctly learn things for the high level because there are only two states, one action, general action plan, right? And it will take a long time, meaning many iterations, to learn appropriately the values, the cues, for example, for the hierarchy, for the bottom of the hierarchy. So naturally, you get a separation of timescales. The general action goals, you learn the values for those, you learn fast. The specific action plans, the values for those, you learn slow, right? And then if you also propose that information from the top levels is provided to each lower level progressively, right? Via value estimation. Okay, I'm hand waving. Then what you get is that you start getting drug-associated accumulation of error at the bottom of the hierarchy, and it goes away at the top of the hierarchy, right? And if you propose that, the level that controls the behavior is the one that has the most sort of dopamine signal on any particular state action opportunity, right? So what you will see is the control being progressively driven to the lower stages, and we imagine that these lower stages are somewhere here, and the higher stages are somewhere here, right? And so that sort of gets you a progression, right? And it's a slow progression, but it doesn't explicitly involve any slow timescale processes, all right? Okay, but let me sort of go back to the slides, okay? All right, okay? So what we've seen so far are these positive reinforcement models, okay? So the drug effect in these models all drives learning, okay? They're good at simulating the initiation of drug taking and the various behavioral aspects like resistance to punishment, blocking versus not blocking, et cetera, et cetera, et cetera. But what they're not good at is giving you a mechanistic explanation, right? So what actually changes? What circuit level processes happen as this pathological learning onsets? And this is what Christian works on and Garrett work on. And so that so these models can summarize certain aspects of that data, but not give you sort of specific cell-based mechanisms, right? The important thing is that, for me, is that these models really can't simulate the pattern of drug consumption. If you put your animal with the lever that gives it cocaine with a certain dose, what's the pattern in time, right? How will the rat actually consume this drug, okay? They can't do that. It's not in the purview. There is no explicit time in these models, right? Okay? No role for withdrawal, right? At least not in the models that I've showed you. Okay, but there is a different theory for reinforcement. That's the negative sort of theory of motivated behavior, and that led to negative reinforcement models, right? And basically all of these models harken back to the opponent process theory that, nicely put forth by Solomon and Corbett in '78 and before by others and exposed later by others. Okay, and so and the story when applied to the drug is the following. Okay, when you take the drug, it drives a positive process, right, that gives results in a positive hedonic state, so a state of euphoria, what I think Solomon called the A process. It's rapid, right, and it's positive. Okay, now after the drug, this positive effect goes away, right, and you get withdrawal, which is an anhedonic state, so a negative hedonic state. Okay, the animal correctly identifies that it feels bad, and so it will do something to make it feel good again. So redress of withdrawal, redress of this anhedonic state. Okay, it's going to go back and take the drug again, and if that loop goes through sufficiently few times, it's not an enormous amount of exposures, but after some exposures, this positive process gets increased, so you get sensitization to the same dose of the drug, right? So then there is this kind of a positive reinforcing bias, right? And then in the long term, right, with the prolonged use, what happens is that, whatever sensitization to the euphoric or hedonic effects of the drug may still be there, or they may decrease. But importantly, what's driven to increase is this opponent opposite process, this anhedonia process, or the B process, right? Or the worsening withdrawal process or craving, okay? And the whole thing about the animal or the addict going through the patterns of drug consumption is, in fact, avoiding this state to go back into this state, which then wears off in time. The drug addict finds himself or themselves again in the state, and they go back to redress the imbalance, okay? So the whole drive here is driven by the negative effects of the drug, okay? the goal, right, is to balance things out. Yeah.
In this particular model, does the baseline state of no euphoria, no hedonic sort of imbalance, does that baseline stay the same? Unlike sort of, because Coop's theory of allostasis shows that progressive decrease in the baseline. So.
Yeah, right. I mean, maybe this cartoon is not quite appropriate to reflect that, but I think that this deepening of dyseuphoria is, can come about because of your sort of baseline expected motivational or hedonic state changes, goes down, and so, or it goes up, and when you remove the drug, right, you go back, you deviate deeper into despair. Okay, all right. So now the point is that the first sort of take on this sort of a process was hedonic allostasis, in the sense that you feel good, you feel bad, right? The redress is all about hedonics, right? So, George Koob and others since then have proposed that actually, this opponent process idea can apply probably shouldn't apply to hedonics as much as should apply to motivational states, right? So they talked about reward sensitivity, right, or the potential of the drug to apport rewards, right? And when you start talking about rewards, you talk about motivated behavior, which may or may not be the same as hedonic effects, right? Okay, so that's an important thing. All right. So, yeah, and so just as you were saying, so the idea is that the reason why the addict descends into the quagmire of addiction is that there is a slow, drug-driven allostatic process that progressively as the addict goes through these cycles of A process, B process, right, it changes the baseline, and eventually the drug addict find themselves into persistent anhedonic or negative process state, and so they are continuously driven to seek and take the drug to try to bring them back up. Okay? All right? And so, and the behavioral paradigm for that was developed, as Marina was telling you earlier, is this dose escalation, right? Short access, animals titrate their pattern of cocaine injections, for example, for cocaine at some level. If you now, same cocaine dose, give them a longer access, right, they start to escalate, okay? If you only had this process, right, if you only had that, okay, you would think that whether you expose them long-term, short access or long access, things shouldn't change, right? Right? As long as the rat is keeping its mean level of cocaine in its body the same, everything is hunky-dory, and whether it's, for 1 hour or 6 hours, right, it shouldn't be different. And if you actually think that this B process is long enough to go overnight, for example, right, and you put the rat back into the chamber again overnight, then the prediction from just that would be opposite of this, right? Because now the rat has loaded up on cocaine enough so that it should get it through the night, and so when you put it again in the same chamber, it shouldn't run towards the lever because there's still some drug left, yeah, or slow drug effect left. Okay, so really, in order to get this to sort of conceptually explain this escalation, you need to think about this slow increase in a kind of a baseline level, right, which then requires more and more and more drug to bring you back up. And this is what they're suggesting is happening. And the pattern of drug taking now, if you look at a single session, has a very specific and stereotypical pattern. That is, there is a loading phase where the animal loads itself up with a drug up to a particular level, one would suggest. Then there is a break, and then there is a pretty regular pattern of lever pressing or administration of the drug. Okay? All right? Okay, and the idea is that under unlimited access, you get escalation, uncontrollable seeking, taking, resistance to punishments, et cetera, et cetera, et cetera. All right, so, all right, let me skip that for a bit. Okay, so now what they also did is they also studied how does the reward threshold change. So this is with intracranial self-stimulation, right? And they found that for the long-access animals, as the consumption of the drug goes up, so does the reward threshold goes up. What that means is that the animal becomes more and more and more sensitive to the absence of reward. Okay, you can think about that, right? Or it becomes less and less sensitive. How does it go?
It needs stronger stimulation.
It needs stronger stimulation, means that, right, means that it goes further and further and further away from some threshold that it requires to identify itself in a normal rewarded state. Okay? So, which would mean that you would, if you keep the dose of the drug the same, you would need more and more and more of it to reach the same state. Okay? And so what Serge Ahmed and George Koob did is they decided to take some of the ideas coming from control theory and put that into a model and see if they could actually, simulate this pattern of escalation, also pattern of drug taking, based on the ideas that you get positive and negative processes and you get a slow negative control adaptation. All right, so let me do this. Let me quickly go through this. So what do you need? What sort of parts of the model that you need in order to do this, right? Remember, there's explicit time here, right? So you need something about the time effects of the drug, okay? So that you can do with a pharmacokinetic model of the drug, right? And so the simple one, classical ones, is a two-compartment PK model where you have the blood compartment, you have the brain compartment, right? There are rates by which the drug enters from the blood to the brain, across the brain-blood barrier. Wow, I said it right this time. Usually I blah, 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 okay. Right? And then there's a rate of elimination back into the blood, okay? These rates are controlled by these parameters, by these variables alpha and beta, okay? All right? There's a constant that you can determine, okay? And basically this model gives you these prototypical alpha function-like time courses for the drug with a fast onset of concentration of the drug in the brain compartment after injection and then a slow elimination, okay? All right? So then you need a model that tells you how does this concentration of the drug affect your functional variable, right? In the case of this particular model, right, you want to, this particular framework, you want to model for how a drug concentration can be converted into the reward threshold, right, or the rewarding outcome of the drug. And that you do with a pharmacodynamic model, which basically tells you how the brain concentration of cocaine is converted into the reward threshold. Okay, and so here is a simulation of an outcome of this change in the PK model, this change in the concentration. This is the effect on the reward threshold, right? So the rewarding effect of the drug is reflected in the decrease of the reward threshold. Okay, so now you have this PK-PD model of cocaine action. You can look in pharmacology journals. This is a classical approach. You can look in the book Computational Neuroscience of Drug Addiction that I edited. There is a chapter on modeling, drug taking with these models. Okay, very classical. Okay, so now you got this. Now you need to do something about the behavior, right? You need to have a model that controls drug taking, right? So what you can do is you can incorporate this into a negative feedback control theory model. So what is that? Okay, all right, the output of the PK-PD model now is the reward threshold, okay? All right? Another variable that you keep track of is the baseline, right? The baseline for the reward threshold, okay? Now you have a sensor somewhere in the brain, okay, that senses the baseline and this reward threshold, okay? Could be signaled by changes in activity in the dopaminergic structures, right? You know, invert this, right? And you can maybe say that this is the dopamine signal, okay? So the signals from the sensor go to a comparator, okay, where basically you compute the error between the set point, this is where you want to be, okay, and this baseline variable that results from the reward threshold and what you had before, okay? All right? Okay, and then this error is sent to a controller, which in fact senses whether this difference is sufficiently large, and if this difference is sufficiently large, that triggers drug delivery. Okay? All right? Okay, notice that this is all in the language of control theory. There are no brain parts here, right? Okay, so it's kind of it's a fairly abstract model, but it's reflecting the processes that are going on. Okay, so now if you take this model, if you now expose it to particular doses of cocaine that come into this PK-PD model, right? What you see is that if you start out at a particular level of reward threshold, you in fact, the goal of this controller will want to decrease this deviation between your reward threshold and the set point reward threshold. It will do that by administering itself with cocaine, right? Because it's far from the set point, it is going to load up, right, to get a particular cocaine dose, to try to get down here, okay? All right? But because of the slow timescales in the PK model, right? Okay? Right? The controller is unable to perfectly titrate how much drug it's going to take, and so it's going to undershoot this sweet spot, okay? And then it's going to stay down here, and it doesn't need to self-administer any more drug because it's below this reward threshold. It feels good, it's rewarded, it feels like it's gotten outcomes that it needs, and that's where this timeout comes in. That's why it's there, okay? And then it will get back close to the set point, maybe overshoot it a little bit, it's hard to see there, and it will take the drug again, and that one singular dose is enough to get it down, right? The PK-PD model will clear the drug out. That will recover the threshold, another hit, another hit, another hit, et cetera, okay? All right, and so there's always this interplay between reaching this and overshooting a little bit, going down, et cetera, right? Okay, and the reason why there is this temporal structure, again, is because there is this timing structure in the PK model, basically, here, right? So the only time is in here, in the PK model, right? So the whole thing that drives the pattern of self-administration is the pharmacology of the drug in the brain and the difference between the optimal reward threshold level and where you're at, okay? All right, so what if you now start moving. So there are a couple of things that you can move in this model, right? What can you do? You can change this PD model and say that the same dose of the drug gives you more hit. Yeah? Okay, so sensitize the animal's brain to the drug, okay? Or you can move this TS, right? Or T0, okay? right? And that, if you move TS, for example, slowly as you take the drug, that means you're changing the overall threshold sensitivity of the animal. Okay? All right. So, yeah. And that's what they went for, and they suggested that as you go into drug taking, right, what happens is that you shift the TS relative to your T0, or vice versa, shift the T0, right? So you overall change the sensitivity of your system to rewards, okay? All right, so that if you start out with a particular frequency of self-administrations and control condition, after you go this allostatic shift in the basal reward thresholds, right, the frequency of your self-administration goes up, dramatically. Okay? All right? Okay. Maybe I have a picture of this. I hope I do. And that not only does the frequency go up between these two situations, but also if you now integrate how much cocaine you've taken here versus how much cocaine you've taken here, you will see that the overall dose went up. Okay? All right? So now the other thing that you can define is you can say, okay, what we're going to do now is we're going to keep the whole thing constant for now. And what we're going to do is we're going to change the dose of the drug. Okay? And what you find is that there's this characteristic relationship that as you decrease of the dose, the number of injections per hour goes up because you need more of the drug to get you down below the reward sensitivity threshold. But then there is a critical dose beyond which you don't self-administrate anymore, right? And that is within the sort of parameters of the model and within the length of the session, okay, you cannot get down below the threshold, right? So they imagine that this is kind of the decrease in the dose is an increase in the effort because you have to press more or run many more times, and so you get this threshold, the break in the self-administration threshold. Okay, all right, and minimal dose for drug taking. Okay, which is something that is observed also in animals. Okay, and so now what they did is they said, well, let's think about the two different kinds of manipulations that long-term drug taking could bring about. One is this change in the sensitivity of the reward, meaning shifting this whole response curve because you shift the basal level, right? Or reward sensitization, and that means that the same dose of the drug gives you more and more transient decrease in reward sensitization, meaning that the rewarding outcome of the drug dose increases, or it could decrease. I mean, it changes. All right, and so they asked the question: how does that affect this curve? Okay, and here's what they found, and that is if you change the baseline levels, right, the minimal dose does not change appreciably, okay? But what happens is that the amplitude of this curve goes up and down, meaning that as you increase the reward sensitivity, move yourself further away from the optimum point, actually the amplitude of this curve goes up, which means that for the same dose, you're going to take many more sessions, right? And that's sort of escalation, okay? The rate of drug taking goes up. If you now do this drug reward sensitization without shifting the baseline, what happens is that you don't appreciably change the shape of this curve, but what you do is you shift it to the left or to the right, whether you increase or decrease the reward response per drug dose. So what you do is you decrease the minimum dose, so you decrease the effort required for the onset of self-administration, right? But you don't appreciably change the number of self-administrations for a particular dose, okay? And the argument from Ahmed and Kub was that, reward sensitization is not what can explain escalation, but baseline sensitization or baseline allostasis can, okay? And, you know, this is sort of a shot across Berridge's bow, you know, and says, look, the way you think about drug effects can't explain our data, although they didn't really say that in the model in the paper explicitly. Okay. All right. Okay.
But
Yes.
When they were going through all this, I mean, all of the, remember, sorry, the incentive sensitization stuff was based on non-contingent drug administration.
Yeah, okay.
So it should have all been over before any of this started.
Yeah.
So why wasn't that part of the grade?
What do you mean it should all be?
Well, the animals should have been sensitized before they
Before they even,
started to.
yeah, I don't know.
Plus the fact that all these things are happening in different brains.
Of course. But, okay, but remember so they had Serge and George Koop did have a neural interpretation of this functional model, and that was that, overall, either dopamine levels or the overall reward sensitivity impact of dopamine, which could be the ratio of availability for D1 versus D2 receptors, for example. That's what's underlying these long-term adaptations, right? Now, for this controller model, they really didn't go into which parts of the brain are subserving which function. Okay, all right. Now, let me sort of say a couple of words here. So unlike the reinforcement learning models, right, this model is very good at reproducing or modeling the pattern of self-administration in time, right? And it can sort of gives you a proposal for what should change for the escalation, right? And all right? Okay, all right. What it doesn't address is how do you learn this behavior in the first place, right? How does the animal acquire the fact, learn that in fact the drug will, lead to a decrease in the sensitivity, particularly if it has a choice, right? So the behavioral paradigms that classically were done for escalation is that it's a kind of a go, no go, right? The choice is you sit here or you press the lever, right? And rats are curious things, they will press the lever, okay? All right, but if you have a choice paradigm, this model would not be able to handle that by its very design. Okay, so you have a kind of a trade-off between the different modeling approaches, right? There's the reinforcement modeling approach, which tells you nicely how it could be that you would acquire and learn drug-reinforced behavior, and you have this negative reinforcement model, which tells you very nicely or proposes very nicely how it would be, why you would have a certain pattern of self-administration to keep yourself at a sort of a normal hedonic level, okay? All right, so this is what I was going to sort of summarize, finish this sort of, the summary of the models part, which is sort of running almost to the end of the lecture, I suppose. So the positive reinforcement learning models, the goal is to pursue maximal rewards, okay? And presence of the drug alters that process. Okay, you can talk about it whether game theoretically drugs make that process suboptimal, or drugs could be seen as just yet another reward, and particularly strong rewards, so that this decision process is still, normal in some sense, right? So it's still rational. Okay, that's a debate. Okay, and there's a sort of a list of some models that some of them I talked about explicitly, some of them I didn't have slides for, okay? And so they're great at understanding precisely how the drugs change the learning process. So they're, okay? The negative reinforcement, there's sort of homeostatic regulation models or allostatic regulation models, So the overall goal of behavior is to optimize organismal stability, and drug alters that process. Okay? All right? And there's actually very classical and large literature on that, and you know, I can't list all the papers here, but at least some of them. All right? And you know, they're great at understanding patterns of drug taking and reward thresholds, but they don't address how you get there. Okay? So the question is, you know, can we come up with a synthesis of the two and reconcile the two goals of behavior? Okay? All right. And so I have a little less than 45 minutes, right?
Yeah, 43.
43 minutes, okay. So in the last 43 minutes, and I won't get to the third part of my lecture, because I was supposed to have 3 hours, now I have less, okay? I'm going to tell you about... Okay, all right, sorry, oops. I tried to slip that one in, but you guys noticed. Okay, all right. What I'm going to I'm going to try to describe to you is our recent work where we try to do exactly this. We try to reconcile or put together the two goals, the goal to maximize rewards and the goal to optimize organismal stability. Okay, and of course, if I was not going to tell you about it, I wouldn't put this question up, right? You know, unless it was the last one, right? Yeah.
Can I just clarify what I said before, because I think the timing matters here, that when I'm not contesting the mass of evidence that prove and, you know, at all have a mass for powerful changes in the HPA axis, you know, definitely changing the reward threshold.
Yeah.
All I was saying is that it's not really a shot across, it's not a good shot across Heritage Bio, because the way they really do these experiments is they start with short access, get everyone trained, and then they take...
Yeah, okay, so the sensitization is already done.
The sensitization is so done, you know, that you wouldn't expect it to, you know, to play out during the escalation phase.
Yeah.
You know, I think it's important to note that. And then, you know, there's really this lovely paper from People's Lab about escalation and activation and different responses in the incumbents that just pulls these things apart, you know?
So maybe I should track back from that. I know that I'm being recorded, so that's it. I'm going to run out of this. I'm going to get run out of this field, tarred and feathered now. But it to me, you know, it sort of felt like an attempt at a sort of a critique of that idea. Okay.
It was, but it was not well conceived.
Okay. Oops. That you're being recorded too. Okay. All right. So let's go for it. Let's do, you know, now I have 40 minutes. And this work was actually done by my former student, Médicale Amati, who is now at UCL. Okay. And we started out from a basic question, and the question was, what is reward? You know, Mehdi came into my lab, and he was a reinforcement learning guy, and he was like, yeah, we just got to modify this reddish model, and it'll be fine. And I kept bugging him by saying, like, yeah, but what's reward? You know, how could we define reward? And so eventually we hit on the idea that, you know, reward really depends on the internal state of the animal, and you have to titrate how you treat outcomes, whether they're rewarding, neutral, or punishing. You have to titrate them against the internal state of the animal, okay? And we know that at least anatomically there's a sort of a potential substrate for exchange of information between homeostatic variables and motivational signaling machinery. And particularly we know that from at least, you know, here part of the homeostatic pathways in the brain that are related to the lateral hypothalamus, et cetera, we know that there are projections to the ventral tegmental area, right? For example, RxNogenic projections from lateral hypothalamus. There are inputs of various kinds of satiety and hunger hormones like leptin, insulin. There is also inputs from temperature-sensitive neurons. Okay, so we know that at least there's got to be, you know, it's likely that there is a communication between homeostatic signals and the dopaminergic signals that are somehow signaling motivational salience of stimuli. Okay, so we decided how could we sort of abstract and formalize that. You know, when you do models, this is what you do. You say, this is marvelous. We know so much about all of this. And there's so much these little errors over here in letters and colors, and it's so beautiful. Okay, so now we're going to forget all about these details, and we're going to formalize it, okay? Reduce it to what we can handle mathematically. Okay, and so let's try it and say that there are certain variables that all of this machinery tracks and tries to get them to be at a particular optimal point. You know, whether you call it set point, like in classical homeostasis, or optimal point, like in allostasis. Let's say that the organism is good when it stays at a particular level. And, you know, for example, you know, we want our organism to stay at a particular temperature, right, 36.6, and not deviate more than 10 degrees up or down, right? Because if you do, you're dead. Okay? And, you know, we want to control glucose levels, blood glucose levels, right? That are also potentially less tightly controlled than temperature, but are in fact controlled. Okay? All right? And what that means is that there is some optimal point for these two variables. So we'll just focus on two because it's easier to draw stuff. Okay? But let's say that you're not at that optimal point. Okay, so you're deviated from the optimal point. Okay, let's imagine that in this homeostatic space, right, that we can define a drive function, right, and this drive function converts this physiological deviation from where you want to be, right, to some signal we call drive, okay, that is, let's imagine, furnished to the motivational machinery, okay? All right? Now, this drive function is nothing but other than a mathematical metric, so a distance between here and here, okay? Now, it could be Cartesian distance if m is 2, n is 2, yeah? Or it could be some other distance if we choose m and n differently, right? For example, under certain choices for m and n, we could have a drive function that is not circular, but is in fact, Jesus, what's going on here? Too much technology. There we go. Okay, so we can have a drive function that expands and compresses differently and in different relative parts of the space. Okay? All right? So far, so good. This is classical stuff, right? 1940s. Okay? All right? Now, let's imagine that we take an action that results in the change of the internal state, K. Right? Okay? So that takes us from the initial state H1 of t, H2 of t, to some new state, okay, right, which let's imagine brings us somehow closer to the optimal, okay? So now we can compute the drive here and the drive here, okay, and we can compute the difference between the two, okay, all right? And we can say that the reward function is going to equal to that difference. Okay, so the reward function tracks how much an outcome of the action we have taken changes our internal, changes the distance in our internal state from the optimal. Okay? All right? So now, any action that brings us closer gives a positive reward. Any action that takes us further away is going to give us negative reward, right? Okay? All right, so we've defined reward and punishment. Interestingly, any action, you know, let's say that we start here and we take an action that brings us here, that's positive. Now we take an action that has exactly the same outcome. Oh, it put us here. That actually moved us away from the set point. That means the same outcome could actually have negative valence because of overconsumption. Okay? All right? Good. Okay. Which means that the reward function depends on the internal state. Yeah? Okay, the same dose of outcome, right, could be rewarding or punishing depending on the internal state. Okay? All right, so now we're going to put this reward in our dear friend delta equation. Okay, and then we're going to update our Q values with that delta. Okay, all right? Okay, so now we have homeostatic regulation, which defines reward, reinforcement learning that defines the values that drive the action selection. Right? So we put everything together. Now, okay, so you have this sort of you can define a nice homeostatic surface space, right? And for any action that you take, for impact of any action that you take within the space, right, you can define the R and you can learn the Qs. All right? So what's going to happen is that you're going to learn the Qs for those actions that decrease the deviation. Those will go up, right? So you'll promote them. Those actions that increase the deviation, R will be negative, right? You will decrease the Qs. They will be much less likely, right? So by this algorithm, the policies of actions that you will learn are the ones that minimize deviations from the homeostatic set point or the allostatic optimum. Okay, as I've sort of told you before, yeah, so here we go. Okay, so K is the outcome, H is this deviation. So I've gone over this. Okay. All right, so the definition of the primary reward is the reduction in the drive. Okay, and then it's interesting that within this framework, you can show mathematically by rearranging things, doing algebra, that in fact, for the Q value for any state action pair, right, is actually online modulated by your internal state. Okay, so you get long-term learning of values, and you get almost real-time modulation of them by the internal state.
How does this account for, sort of in the short term, an action made that wasn't driven? So just trying, just trying.
Oh, okay, so one thing is that the action selection that happens here still goes through this softmax equation, which means that if the max is soft, right, then you have a proportion of your actions that are exploratory, right? And so you take them and evaluate them, and then progressively you learn them. Okay, so that we haven't actually, it's another aspect of this theory to work on. Okay, all right. So now, but as I told you before, okay, this is all very nice and lovely and whatever, right? But what if it screws up the optimality of this reinforcement learning algorithm, right? You know, I've hinted to you that from formal machine learning or economic approach, reinforcement learning models are really great because they guarantee that you will eventually converge to the optimal rational behavior, right? That you will seek and gain the most toys you can possibly gain in your lifetime, right? But here we've just defined this reward kind of not from those principles, okay? So well, the nice thing is that what we showed mathematically is that if you use this learning algorithm and to structure your action policies, okay, the action policy that minimizes the sum of future discounted deviations is also the action policy that will maximize the sum of future discounted rewards. Okay, so we mathematically showed that in fact physiological stability, right, and this sort of economic rationality are one and the same for this reward definition. Okay? So, you know, this debate of whether animals strive to preserve their homeostasis or strive to collect rewards really doesn't exist within this framework, right? Okay? In other ways said, our biology is designed to make us economic performers in some way. Or, our economic performance really is geared towards ensuring our physiological stability. Okay, all right, that's great. So there's the proof. It was actually a theorem, whatever. Okay, now what does this have to do with the price of potatoes, right? Meaning like with this course, which is all about addiction. Okay. Well, first thing that we did is we said, well, let's see if we could, try to at least simulate some classical experiments on the drug-induced adaptations. Okay, and one of the classical experiments was these experiments that were done in the 60s and 70s on understanding the mechanisms of alcohol tolerance. Okay, and I don't mean specific neural mechanisms. I mean the general organismal mechanisms. And so the setup and the question at that time was that is alcohol tolerance learned, or is it sort of a pharmacological adaptation of body tissue? All right. And the assay that was used was to look at the hypothermic action of alcohol. Right. So if you inject an animal with alcohol, it becomes more sensitive to temperature deviation. Okay, all right, so which means, if you're out there skiing, having a bit of schnapps won't make you warmer. It'll actually make you colder. It might make you think that you're warm, but anyway. Okay, and so the setup was the following. All right, they had a rat or a mouse, I think it's a rat. There was a bell, right? And then there was a temperature deviation. They were cooled down with water, right? And what they looked at was shivering. Right? Did the rat shiver or not shiver? And how much did it need to be cooled down to start shivering? Okay? All right? And so the experimental data was the following, right? So they looked at shivering. They also looked at body temperature, actually, directly, right? Measuring body temperature. And so what they found is that, relative to control, when they injected the rat with alcohol, they found that initially their body temperature was deviated relative to control rather strongly. So there was a strong effect of the drug. Okay? All right? And then as they progressively repeated these sessions, okay, they found that this deviation decreased. Right? So this is kind of a classical assay for adaptation to drug effect. Okay, so it's a physiological effect, all right? So it's not about wanting or liking, it's about, being cold or not. And obviously, you may think that the rat doesn't like being cold, and et cetera, et cetera, et cetera, right? So interestingly, what they did when they did a catch trial, where they did everything except inject the rat with alcohol, right? When they cooled it down, or even before they cooled it down, right? They found that the rat's temperature went up here. Right? As soon as the bell rang, the rat's temperature went up. And then when they did the next trial where they again injected it with alcohol, they saw that there was a negative deviation again. Okay, all right? Yeah, and so this is the key trial. Okay, all right? So in order to model that, what do you need to do? Well, you need to have some kind of a model for the time course of alcohol in the body. So this is kind of like a PK, cheap man's PK version, PK model of alcohol, which we just wrote down, okay, and adjusted the time course according to the data that was available in the literature. So this is what happens directly to the temperature when you inject alcohol. Okay, we further hypothesized that there is some kind of a tolerance response, direct tolerance response to this alcohol that develops in the rat's body, right? And the question is, does this tolerance response become bigger, for example, as you go down the timeline of this experiment, or is there something else going on? Okay? So, you know, if the tolerance response becomes bigger, right, then alcohol tolerance is not learned, it's just an adaptation, right? It's an adaptation of the internal machinery, okay? All right? But in our model, this tolerance response didn't change with time. Okay, it was kept constant. This was kept constant, this was kept constant across sessions. Okay, and so the total effect of alcohol directly was, you know, this minus that. So you get this kind of a biphasic change, well, sort of hypothesized change in the temperature. Okay, so now you're going to, let's see, write down the state space for our experiment so that we can put it in the Q-learning algorithm. Okay, so you have a S0, which is the presence of the bell. Okay, and then you have two potential states. One is where you develop a tolerance response, and one is where you don't. Okay, so this would be injection of alcohol, this would be not, right? And then there are states that track explicitly the different time points within the experiment. Okay, and then eventually we restart. Okay, so that's the state space of our Q-learning model. Okay, and I wouldn't go through all of this if it didn't work. Okay, so these are the simulations of the model. Okay, so progressively from session to session, you see a decrease in this change in the temperature under injection of alcohol. What's neat is that for free, somehow it fell out, well, because we had explicit timing information on the alcohol and the tolerance response. The actual pattern of these deviations for each session looked like the experiment. And then when you presented the catch trial, you saw this anticipatory change in the temperature, right, which was not necessary because there's no alcohol being injected, right? But the reason why it happened, right, is because our reinforcement learning model learned that it needs to generate this the positive change in the temperature, all right, in anticipation of the negative deviation. Okay, now the tolerance response in our model was kept constant, right? So actually, what this suggested is that, you know, in these experiments, it's not the tolerance response that developed, right? It's that the rat was conditioned, right, to produce an anticipatory change in its temperature, right? Okay, so alcohol, so this suggested, in fact, that alcohol tolerance is a Pavlovian learned anticipatory response. Okay, all right. Okay, I have 20 minutes. Okay, so next, what we wanted to do was to apply our model to the escalation paradigm. Okay, why? Because we felt that we've got all the pieces necessary to deal with escalation. And not only with escalation, but also with acquisition of escalated behavior, right? Learning. Okay, and so basically, yeah, I don't have to go through the paradigm again, okay? And the suggestion that we're making, and I'll show you some results of the modeling, is that this progression to addiction, if you think that escalated animals are really addicted, is really a pathology of homeostatic regulation of reinforcement learning. Okay? All right? Okay, and so again, when you first start, what you need to do is you need to model the state space of our task, where, you know, you can press the active lever to get some cocaine, okay? Or press the inactive lever and stay exactly where you are. You know, motivationally, if you press the active lever afterwards, you can press the active or the inactive lever, et cetera, et cetera. So this is sort of modeling that cycle of potential actions. Okay, and then, you know, you need to also include the timing information of what happens when you press the lever and you get a cocaine infusion. So there is a PK model built into it, okay? And the PD part of the model is this drive function, okay? So we now presume that the effect of a dose of cocaine, which has some time course, that's the PK, right, model, okay, affects some regulated variable in the brain, okay? All right? So what regulated variable might it be? What might it have to do with? Duh. Cocaine affects dopamine, right? Okay, so we suggested, just as, not suggested, but basically we said, let's say that there is a regulated variable in the brain that is related to cocaine. This could be overall level of dopamine signaling, right? This could be availability, relative availability of D1 versus D2 receptors, you know, something like that, okay? All right, and that figures into our drive equation, and it's this drive equation that acts like the PD model, right? And the outcome of this PD model, of this drive equation, right, is the definition of what is the rewarding quality of an outcome of the action that the animal takes, right? For example, if the animal takes an action that apports cocaine, that changes the animal along this regulated variable axis, let's say dopamine levels or D1, D2 availability ratios, right, which would move it closer or further from a set point, right, and that will go into our Q learning equation. Okay, all right. Okay, one more assumption that we made. So, and it's not surprising that this assumption was made because Mehdi went to Serge Ahmed's lab and actually ran rats and talked to Serge and sort of went along this sort of idea of thinking that chronic cocaine, what chronic cocaine will do is shift the set point to higher values or to lower values somewhere along this axis, right? Thereby shifting the drive function and therefore changing the rewarding outcome of a particular dose of cocaine, all right? You can see where this is going, okay? So this is fast, this shift is slow, right? Okay, and so now we're ready to simulate what would happen if we put our animal in an escalation paradigm, okay? All right. Okay. So first of all, we can readily simulate escalation of dose. So these are simulations from our model. Under short axis, essentially this slow allostatic process is such that it doesn't build up enough not to relax sufficiently between self-administration sessions, and so it goes away, and on the next session, cocaine doesn't have too much reinforcing effect relative to the last session, right? On the long axis, the animals in fact self-administrate self-administrate cocaine for sufficiently long, right? So that between sessions, right, the set point doesn't go back to its control, right? And therefore, in the next session, right, cocaine has a potentiated impact on the reward, okay? Which means that the value of that behavior goes up, right? Which means that you activate the responding, which means that you take more, right? And it's a kind of a runaway process. And so basically what you do is you escalate here, you don't escalate here. Okay, so this is reflected here. The homeostatic set point goes up in the long axis animals, but not in the short axis animals. Okay, all right. And this is just a blow up of what happens on initially on day 1, day 2, day 3. Now, the point here is that the reason why, the reason why you escalate, the underlying reason, is not because the animal sinks deeper and deeper in some sort of a state of, reward deprived or anhedonic despair, right? The reason why it goes up is that the same dose of cocaine from one session to another has a potentiated impact on the reward machinery, right? So it's sort of like it's a negative process model with a positive reinforcement. Okay? So the animal learns that it's more advantageous to take more and more and more cocaine. Okay? All right. Okay. So now, in this model, we can also simulate loading. Okay, so here's some data on loading. These are infusions at the beginning of the session, pre-escalation. This is after escalation for long access. So let's look at this. So the more you escalate, the more loading you get, right? So at the beginning, this is exactly what we get from our model, these sort of shifts that if you escalate the animals, you get higher loading frequencies. Okay, all right, okay. Same, we can simulate effects of the dose, right? So, for example, if you change the dose in both long axis and the short axis rats, the higher the dose, right, the slower is the rate of cocaine self-infusions, self-administrations, right? This is exactly what you see in our model also. But interestingly, for this paradigm and also for our model, even though you, in both escalated and short axis rats, if you increase the dose, right, they decrease the frequency. And so they do that to titrate the average amount of cocaine they get no matter what the dose. It's just it's higher for the escalated animals, right? And this is exactly what you get in the model as well. Okay? All right. Okay. And so now we can also simulate an experiment with punishment associated with cocaine self-administration, right? So progression to compulsivity, right? And the data, let's just look at this, right? And the data was such that in the non-escalated animals, punishment had a significant effect decreasing the probability of responding. So this is percent pre-punishment responding. So this was, punishment decreased responding to 60% of what it was, I think. That's correct. Okay. All right. Okay. Whereas in the escalated animals, it didn't change anything. Okay. And so you can simulate this situation with this state space or Markov decision process where you add punishment to the active lever. You can add it here, you can add it here. Doesn't make much difference, right? And so you look at responding basically in the long axis and the short axis, and what you find is that the short axis model is exquisitely sensitive to punishment, right? And the long axis model is not, okay? And interestingly, that it will decrease its response on the trial when you actually punish, so it will learn that it should, immediately it will learn that it should avoid things associated with punishment, but on the sessions afterward, it hasn't really learn to associate punishment with this particular behavior, right? Okay. So, Okay. So further experiment that we tried to simulate is non-contingent cocaine-induced relapse. Right? So it's going to hard for me to describe this experiment in detail because I didn't do it, and Mehdi actually got into it when he was in Bordeaux, and so no matter how many tries I try to read this paper, I always forget the details. But basically the point was that escalated animals were then extinguished from cocaine taking, and then some time after extinguishing, they got a dose of cocaine, right? And that reinstated their behavior transiently. If they got another dose, they reinstated transiently, et cetera, et cetera, et cetera. Okay? All right? And so that's also what you get in our model, so that if we extinguish our escalated animal behavior and you give them a dose of cocaine, so the model will start picking or choosing the cocaine action versus the non-cocaine action. Okay, so you get, this kind of reinstatement you can't do neither with reinforcement learning models nor with these negative delayed feedback models, right? And the reason why you get reinstatement is very interesting, that in order for us to capture this reinstatement behavior, we had to do something with our model, and that is we had to presume that in fact the animal explicitly encodes whether it is in cocaine available environment or cocaine non-available environment. And the way you do that in the model is you have these state spaces, these Markov decision processes, but in addition to the values, what you do is you have an explicit model, right, representing the transition probabilities between these states, right? Notice that in the standard reinforcement, model-free reinforcement learning algorithms, this is not in the brain, right? This is your representation of the task, okay? Right? In the model-based reinforcement learning algorithms, this is in the brain. It's learned. It's learned by inference processes, and it could be even learned from exactly the same reinforcement learning area, right? And so in the cocaine available MDP, right, we added, the difference between the two was that in the cocaine available MDP, we added that there was an encoding of specific outcome that was associated with going from S1 to S2, and that outcome was the cocaine, okay? All right, and so the way we were able to simulate this reinstatement is to say that, when the animal is exposed to a cocaine available environment, right, it will reinstate the behavior that it was conditioned to do in that environment, right? How does the animal know it's in a cocaine available environment? Well, it was just given a dose of cocaine. Okay? All right? It was given a dose of cocaine that shifted its internal state, okay? And that was recognized as the cocaine available environment. Okay? So for this particular task, the shift between neutral and cocaine available environment was actually through some variable that reflected the presence of cocaine, right, pharmacologically. But it could be a presence of some stimulus that is associated with this MDP and not with this MDP. Okay, and so the black lines is what happens to our model when it's in this environment, and then we inject cocaine. No, it doesn't quite respond.
Yeah. Can you simulate stress-induced reinstatement or other forms of reinstatement?
Maybe, potentially. I mean, this is our first tack on this model, and we're still struggling to get it through all sorts of pitfalls reviews. And then we got this one review that said, how dare you to think that transition to addiction is a dopaminergic disorder? Okay. No, but anyway, But we haven't. So you could potentially incorporate different stimuli that precipitate reinstatement. But you would have to. We would have to think very clearly and very carefully about how to incorporate that into our model. Okay, to go beyond sort of overall kind of semi-triviality, to say anything that shifts the internal state significantly, right, will signal to the animal that it's in a drastically deviated state, right? Because that deviation also has to be along the right dimension in the homeostatic space, et cetera, et cetera. So it will take us some time to figure out how to do that. But I'm hopeful that we'll be able to do it.
Quick question.
Yeah.
It's really cool. Did you, was the paper you were using to base your model on, did they do extinction training before reinstatement?
I don't remember.
I'm just wondering if your model is at this point completely independent of, whether there's been any intervening state of the animal before it reinstated.
Yeah, it's a good question. I have to go through the details again. But you could. So basically, what controls you between this state and that state, which then controls how well you would reinstate or not, right, is this variable C, right? And you could propose that there is a slow dynamic for C as well, right, which allows you to progressively, differentiate cocaine available versus cocaine non-available environments. You could also think about pathologies of this C, that if this C goes, so it's a 0, it's no cocaine. If it's non-zero, it's cocaine. The structure of the state space is different. So let's imagine that something goes completely wrong and the C goes towards 1 all the time, right? Well, that means that our model will I don't want to say word think because that's anthropomorphizing our model, but it'll be in a state where it infers that it's in a cocaine available environment. Okay? So now when cocaine is not available anywhere anymore, right? I wonder what that would imply for the kind of structure of behavior that you could get out of this model, right? And there's lots of stuff to do still. So it's really new, right? Now the thing is that if you need this sort of explicit encoding of state outcomes and action outcomes in order to take care of these critical points about addictive behavior, right? And it's exactly the same model that was reproducing the escalation, and part of the escalation was changes in the C, right? Then we can claim that, in fact, that maybe progression to addiction is not a transfer from goal-oriented to goal-independent control of actions, right? But it is a pathology of the goal-oriented system, so that, and this is a kind of a, so this is a kind of we'll stop there because I think it's time to go to the beach. Yes. All right, I didn't get a chance to tell you about nicotinic receptors, but well, over a beer and a cigarette sometimes. But generally when you think about what goes wrong in your motivation or learning processes when you become an addict, is it that your learning algorithms are normal, but you're operating on the wrong data? You're just not. You're getting reward information that shouldn't be there, like, the drug, okay? Or is it that the drug subverts the learning algorithm, right, in such a way so that even when you are operating on the correct data, meaning that there is no drug anymore, right, you will not learn the correct behaviors, right? And so there's always, in my mind, it's always this question, which one is it? Right? The answer in biology is probably both, but the question is under which condition one plays a bigger role than the other. Okay? So here, for example, you know, you would say that if the C is subverted by the presence of the drug to be always such that persistently the animal is in this MDP or state space, it believes where the cocaine is available, right? It's sort of the latter. No, it's sort of the former. It's sort of different, right? So it's what happens is that not only the learning is different, but in the end, the animal's worldview is different, right? So it's not that the addict becomes habitualized, it's just that their estimation of what they can do is completely different, or what they should do is completely different, right? So yeah. So I'll stop there, and we have 15 minutes to get to the bus. Thank you.